So let us discuss about cell injury too. There are several causes of cell injury. One is oxygen deprivation. Oxygen deprivation can be due to hypoxia as well as ischemia. Hypoxia is reduced oxygen supply to the tissue. For example, in anemia, decreased hemoglobin and decreased oxygenation of tissues. While ischemia is both oxygen as well as nutrients are compromised. Blood supply to an organ is blocked by a thrombus or something. Not only oxygen is reaching the not only oxygen supply is compromised, but also the nutrients such as glucose is compromised. Then physical agents, extremes of temperature, for example, deep burns and cold. All these things can cause cell injury. Mechanical trauma can cause cell injury. Then chemical agents and drugs. Carbon tetrachloride poisoning can cause cell injury. Arsenic poisoning can cause cell injury. Infectious agents, submicroscopic viruses to large tapeworms. Immunological reactions, genetic derangements, nutritional imbalances, undernutrition as well as overnutrition can cause cell injury. Now, let us see the mechanism of cell injury. There are several mechanisms of cell injury. One is depletion of ATP. We know that whenever there is ischemia, less oxygen supply, the aerobic oxidative phosphorylation is compromised. So there is less oxidative phosphorylation, so there will be less ATP. What happens if there is less oxidative phosphorylation and less ATP? Anaerobic glycolysis will take place. In anaerobic glycolysis, the glycogen stores will be depleted. The glycogen will be converted to pyruvate to lactic acid. So there is glycogen store depletion, and the glycogen will be converted to pyruvate and pyruvate to lactate. This lactic acid can cause decreased pH. When there is acidity, there will be decreased pH. And we know that there are rough endoplasmic reticulum, that is endoplasmic reticulum with the attached ribosome. When there is decline of ATP, these ribosomes can get dissociated from the rough endoplasmic reticulum. This dissociation of ribosomes from the rough endoplasmic reticulum can cause decreased protein synthesis. Another problem is, we know that there is a pump called sodium potassium ATPase in the cell. This sodium potassium ATPase will pump 3 sodium out of the cell and takes in 2 potassium. So when there is less oxidative phosphorylation and less ATP, the sodium potassium ATPase pump will fail. So the sodium will be retained inside the cell. We know that water moves from a area of low osmotic pressure to high osmotic pressure. So due to failure of the sodium potassium ATPase pump, there will be more sodium inside the cell. So water moves from the outside to inside of the cell, where there is more sodium, more solute concentration and osmotic pressure. So then when water entering inside the cell, this can lead to cell swelling as well as swelling of the organs, swelling of endoplasmic reticulum, mitochondria, etc. These are the consequences of reduced ATP. Then there are other mechanisms of cell injury. One is mitochondrial damage. When there is increased cytosolic calcium, reactive oxygen species, etc. can cause mitochondrial damage. We know that the mitochondria has an outer membrane and inner membrane. Inner membrane is thrown into holes called cristae. Whenever there is mitochondrial damage, there is formation of a channel called mitochondrial permeability transition channel. These mitochondrial permeability transition channel can leak protons, H+. Leakage of these mitochondrial permeability transition channels of protons leads to loss of membrane potential. Leakage of protons through these mitochondrial permeability transition channels leads to loss of membrane potential, and there will be decreased oxidative phosphorylation. Further decline in ATP production takes place. Another mechanism is they can leak cytochrome C. The cytochrome C is an integral component of electron transport chain. So there will be further decline of oxidative phosphorylation. Not only this leaked cytochrome C can combine with apoptosis activator factor. So this can lead to apoptosis or programmed cell death. So these are the mechanism of mitochondrial damage. Now we will learn about damage by the increased cytosolic calcium. Normally inside the cell there will be less calcium, usually less than 0.1 micromoles. Outside the cell it is about 1.3 millimoles. So outside the cell there is more calcium, inside the cell less calcium. This potential is maintained by a pump called calcium magnesium ATPase pump, energy dependent calcium magnesium ATPase pump. So what happens if there is less ATP? This calcium magnesium ATPase pump will fail. So calcium enter inside the cell. Also there can be release of calcium from the endoplasmic reticulum and the mitochondria. So there will be increased cytosolic calcium. Failure of calcium magnesium ATPase pump increase the calcium inside the cell as well as release of calcium in the endoplasmic reticulum and mitochondria. This calcium activates many enzymes. Phospholipase they activate. Phospholipase will damage the membrane. Then there will be nuclease which will digest the nucleic acid. Then protease enzyme also they will activate. So there will be digestion of the organelles. That is about influx of intracellular calcium. It will activate all the enzymes. Oxidative stress we will learn later. Now we will discuss about the morphology of reversible and irreversible cell injury. So sodium potassium ATPase pump will fail, so that more sodium is retained inside the cell. This can lead to cell swelling as well as swelling of endoplasmic reticulum, swelling of mitochondria, etc. And there can be cytoplasmic blebs, swelling of endoplasmic reticulum, mitochondrium, cytoplasmic blebs, as well as cell swelling. We know that decreased ATP due to aerobic oxidative phosphorylation, anaerobic oxidative phosphorylation will take place. So that glycogen will be converted pyruvate and pyruvate to lactate. So this because of lactate there will be decreased pH. This can lead to clumping of chromatin due to decreased pH. And there can be myelin figures. These myelin figures are derived from the cell membranes of the damaged organelles. Myelin figures derived from the membranes of the damaged organelles. Suppose the injury still continues, there can be severe damage to membrane and the mitochondria. So when there is severe damage to membrane, the lysozyme membrane will rupture. So this can lead to all the enzyme released into the cell from the lysozyme. And there will be severe mitochondrial damage. In reversible cell injury, there can be small amorphous densities can be there in the mitochondria. But in irreversible cell injury, severe mitochondrial damage and large flocculent amorphous densities. Can be appearing in the mitochondria. Density in the mitochondria. This is a feature of irreversible cell injury. And severe membrane damage, so that the lysozyme membrane rupture and lysozyme enzyme can be released. Severe membrane damage and severe mitochondrial dysfunction. This characterize the irreversible cell injury. Now we will see some question. Myelin figures are derived from the membranes of the damaged organ, cell membrane. These are the answers. Myelin figures can be seen in both reversible and reversible cell injury. But in irreversible cell injury, myelin figures will be more increased. Electron microscopic finding characteristic of reversible cell injury. Dilatation of endoplasmic reticulum and mitochondria, that is and cell swelling, that can be seen in reversible cell injury due to increased water entering inside the cell. Dissociation of ribosome from the rough endoplasmic reticulum and decreased protein synthesis can be seen in reversible cell injury. Small densities can be found in the mitochondria in reversible cell injury, but large flocculent amorphous densities is characteristic of irreversible cell injury. Minor figures can be seen in both reversible and irreversible cell injury, but it is more increased in irreversible cell injury. So the answer is larger flocculent amorphous densities in the mitochondria. That is all today. Thank you.